All right, so today we're going to finish up chapter 15, antimicrobial drugs. Specifically, we're going to be talking about antivirals, anti-eukaryotic drugs, and resistance to antimicrobial drugs. Okay, specifically, we will be going over the learning objectives. I'll open it up to questions. We're going to review the different eukaryotic kingdoms that we're concerned about in microbiology, antifungal drugs and their targets. We're going to review viral replication. We'll talk about antiviral drug classes and their targets. And then we'll talk about mechanisms of resistance, what's due by Friday, and the plan for next week. Okay, so this chapter helps us move toward being able to meet the following learning objectives. Be able to define the role of the key role of evolution as it applies to microbiology. Identify microbial structures and connect the structures to the functions. Identify necessary components and processes and microbial metabolic pathways. Identify key agents and processes of microbial information transfer. Identify pivotal components of microbial systems important to human health, and analyze and describe the impact of microorganisms. Okay, any questions? Okay, let me know if you think of any. I'm happy to answer them. Okay, so let's go to the whiteboard. We're going to review eukaryotic kingdoms, specifically those that are important in microbiology. Okay, so just as a reminder, okay, eukaryotes all have a nucleus. Okay. And in addition to that, they also have membrane-bound organelles. Okay. So, what are the kingdoms of eukaryotes? I know it's been a long time, but as you struggle to remember, you'll remember it longer when you get the answer. Okay, let me help get you started. So, we humans are eukaryotes. What kingdom do we belong to?
Animal.
Animal, very good. Okay. All right. And then the next most obvious one is spring, they're blooming.
Plants.
Plants. Okay. And then these guys bloom all the time. Well, bloom, they release spores, but we make good use of some of them in making, things like bread and beer and wine and
Fungi.
Yeah, it's very good. And then which category do we toss everything in that doesn't quite fit into these other three?
Yeast.
Yeast are actually under fungi. Let's go ahead and put under fungi, we have mold, and we have yeast, and we have delicious mushrooms, and poisonous ones too. I don't know if that helps, but if it's not an animal, if it's not a plant, if it's not a fungi, we toss it into the
Protozoa.
Very good, protozoa. Okay, so in microbiology, of course, we're interested in animals because, we are animals. And, we also study worms and different things that, to identify an infection it's, we use a microscope to do it. And of course, we're interested in fungi because, they have microscopic species, parts, okay? And then we're interested in protozoa. Generally, we leave the poor plants out, okay, unless you're a microbiologist that specializes in plants and looking at plant disease or looking at, their microbiome. Believe it or not, plants have a microbiome too, okay? So we're mainly interested, okay, in animals, fungi, and protozoa, okay? So let's go ahead and list some characteristics that make fungi and protozoa different from animal cells, okay? So what's a structure that, say, a yeast would have that we would not have? I know it's hard to remember. On the outside of the cytoplasmic membrane.
Cell wall.
Very good. Yes. Good job. Yeah. So fungi have a cell wall, and what's it made out of?
I don't know.
Starts with a C.
Cytoplasm.
You would think so, but yeah, no. Cytoplasm is kind of gushy. It's made up of chitin, which interestingly enough is the same stuff that makes up the shells of, spiders and grasshoppers and other creepy crawlies. Okay, so why do I bring this up? Okay. Now we last time we talked about bacteria, which are prokaryotes. What do they make their cell walls out of?
Peptoglycan.
Very good. And I did not give myself enough room to write out the entire word all in one line. Okay. So in other words, the antimicrobials, the antibiotics that we talked about, that will inhibit bacterial cell wall synthesis will not affect fungi. Okay. Okay, so this is something that is different. Now, what's easier is to list what all of these kingdoms share. Okay, so what do they all have? We've listed one up here. They all have a nucleus.
Membrane-bound organelles.
Very good. Okay. And what's the structure that makes proteins?
Mitochondria.
Oh, let's go ahead and put mitochondria down because they all have mitochondria. But mitochondria make the bulk of the ATP. But good that you remembered that all eukaryotic cells have mitochondria or structures that used to be mitochondria. And the one I'm looking for starts with an R.
Ribosome.
Very good. And they're all what size?
80s.
Yep, they are 80s. Okay. Okay, we're listing a lot of stuff that's the same. Yeah, there's not a lot of targets for antimicrobials. Yeah. Okay. And so that's part of why we don't have a lot of antifungal drugs, not compared to the antibacterial drugs. We have a whole slew of antibacterial drugs. And it's because there's a lot of differences between bacteria and our cells. Okay? Fewer differences between fungi and animal cells. Okay? Cell wall, that's one of the big ones. Okay? Interestingly enough, we do not have, so far as I know, drugs that target chitin formation. Okay? If you decide to go into drug development, that's something to look at. Okay? Because if we targeted this, would have very few side effects. Okay? But one thing that is different, okay, all cells have a cell membrane. Sometimes we call it a plasma membrane or a cytoplasmic membrane or a cell membrane. All the same thing, pretty much. But there are different molecules in it, okay, that are unique to the different kingdoms. Okay? And for fungi, there is a relative of cholesterol called ergosterol. Okay? So in animals, we have a rather similar molecule called cholesterol. Okay? Cholesterol gets kind of a bad rap, okay? Because we don't want it floating around in our blood and coating our arteries. That leads to hardening of the arteries. Okay? But cholesterol is important to maintaining the fluidity of the cell membrane. Okay, because the cell needs to be able to move around without popping that cell membrane. Okay, so cholesterol helps it hold together. Okay, especially as things heat up, and then as things cool down, it keeps your cell membranes from setting up like butter. Okay. Okay, so we actually have drugs, antifungal drugs, that target synthesis of ergosterol, okay? Or it'll target ergosterol directly, okay? So if I have a cell membrane, okay, here's my lipid bilayer, okay? And I've got some ergosterol. And it's a steroid, so it's this multi-ring structure. Okay? And I'm only going to draw 2 rings, but there's 4. These drugs, okay, will bind to them and kind of put them together so that it creates pores. Okay, so if I create pores, what's going to happen to that cell?
It's going to explode.
Yeah, it's going to explode. Okay, it's going to lyse the cell because I've got all sorts of water flowing in. I've got all sorts of stuff flowing out that we don't want to flow out. Okay, and it causes the cell to pop. Okay, now we've got some other ones. Well, we've got quite a few antifungal drugs that will target this particular molecule in the cell membrane. Yes, Michelle.
Okay. So you're saying that a difference between fungi and what is that their cell wall is chitin?
Yes.
In bacteria. So, okay. So, but animals don't have that as a cell wall.
Exactly. Yeah, because we do not have a cell wall. So no cell wall.
Okay. Okay.
Now this is quite simplistic, but one way to remember it is plants have cell walls and they don't get up and move around because they're kind of stiff.
Okay.
Yeah, it's kind of like the Tin Man when he's rested. Okay, they can't move around very fast. Okay. Whereas we animals, we're like the Scarecrow and the Wizard of Oz. We can move all over the place because we don't have cell walls.
What about protozoa?
Protozoa, they don't have any cell walls. Well, no true cell walls.
Okay, thank you.
And I am not a specialist in protozoa. There are some that do have, like diatoms have kind of like a glass shell. If you ever look at them underneath the microscope, they're really quite beautiful. But for whatever reason, that's not considered a cell wall. It's more like a shell. I don't know.
So if let's say that they're doing the stuff to go through the cell membrane using this ergostol, is that going to kill a spider, but it's not going to kill bacteria?
Oh, okay. So actually, you're right. It would not kill bacteria because they don't have any, so far as I know, they don't have either one of these steroids in their cell membrane. They use other stuff. So this would probably bother a spider about as much as it bothers ourselves.
Oh,
because spiders are actually animals, and so they use cholesterol in their cell membranes.
But wouldn't it break up the cell walls that make their shells?
You would think so. And the drugs that target ergosterol do not target chitin.
Oh, okay. All right.
Yeah.
Okay. Thank you.
Oh, quite welcome. But I'm glad you brought that up because we do have some problems with some of these drugs targeting cholesterol. Okay. They don't target them at cholesterol as well as ergosterol because they're a little different shape. But we have higher rate of side effects with oral antifungals. Okay. Now, well, having said that, you've probably seen the commercials for a pill to take to treat toenail fungus. That's oral. It's over the counter, so it must not have severe side effects. It must not be targeting ergosterol. I actually don't know what they target in those. Those are fairly new drugs. But what do we do for most fungal infections? Okay, like I've got athlete's foot and I've got toenail fungus, but we're going to ignore the toenail fungus. How do we treat athlete's foot? Fungus the skin between the toes.
Keep it dry and put powder on it.
That's right. Keep it dry, put powder on it. We're taking the water away. We're making an environment where fungi won't grow. But let's say
wear
that's
shower
still not working.
shoes.
Yeah.
Antifungal cream?
Yeah, we put a cream on it. Yeah, we call that topical. Okay, where you put it on just one area of the body, usually on the skin. Okay, topical application. Mouth rinses sometimes can be considered topical because you put it in there, you rinse, you spit. Okay. So with most antifungals, up until they came up with that one for the toenail fungus, they tend to be topical, okay, because we have fewer side effects. Because with these skin fungi that are causing problems, they're eating the dead, dry skin on the outside. And if your immune system didn't notice them, we wouldn't care. They wouldn't be causing a problem. But your immune system notices, and that's why you get the itchy inflammation that's associated. And so, yes, to prevent athlete's foot, it's good to wear, dry out your shoes, dry in between your toes really well, use foot powder, all of these things to prevent it. But, if it doesn't get bad, then we can have some over-the-counter topical medications. Okay? But if with immunocompromised patients, we can see systemic or whole body fungal infections. Okay. So say I've got a patient that's on chemotherapy for leukemia. Okay. Sometimes they can get a blood infection of yeast. Okay. We call it a fungalemia. And it's usually pretty serious. Okay, because you know, if you're immunocompromised enough that you get a fungal infection in your blood, that's not a good sign. You know, same thing with folks who have progressed from being HIV positive to being, you know, into AIDS, acquired immunodeficiency syndrome, or, you know, patients that are on immunosuppressives for autoimmune diseases or an organ transplant. Okay. In that case, we would treat them with a systemic antifungal that does target the ergosterol. And the one I want you to know is amphotericin B. And we only give this in the hospital, okay? It is not something that you would go to the pharmacist and pick up a pill for, okay? We generally give it by IV, and we do it in the hospital so that we can keep an eye on the patients, okay? So that if they start to develop some of these side effects, then we can stop treatment before there's any permanent damage, okay? So for whatever reason, nerve cells tend to be more sensitive. And so if a patient starts complaining of, you know, starting to have trouble with their hearing, okay? Or sometimes we'll see severe vomiting and diarrhea, okay, because you're having an immune response, okay, or anything like that, then, you know, we stop the medication, okay? And the reason why I want to emphasize that is because there's fewer targets. We're too closely related to fungi. And there's just not as many different targets. But they found something for the toenail fungus. I don't know what they found, but they found something. Okay. And it's kind of the same thing with protozoa. Okay. It's hard to find targets for anti-protozoal drugs. Okay. We do have some, especially for malaria. And we're still not quite sure how they work, but they work and most people don't get sick from it. Yes, Sherry.
So the amphotericin B, what are you saying that that's to target fungus?
Yep, it targets fungus. Specifically, it targets ergosterol.
And so what type of fungus would be so serious that you have to put it through IV? Like what kind of situation is that?
Yeah, the one that I'm most familiar with is, like I said, with somebody who's immunocompromised. They can get some yeast infections in their blood. Sometimes it can be the Candida albicans, but sometimes it can be other fungi that started on the surface or in the lungs and then was able to invade into the blood.
Okay.
Yeah.
That's bad.
Oh, quite welcome. Yeah, it's usually pretty serious. Yeah. Okay, how are we doing? You ready for me to ask you questions? By the way, good job on all the questions. You guys have asked very good questions. But now it's my turn. Okay. Let me pull up a poll here. Okay, which of the following is a structure the fungi have, that fungal cells have, but animal cells do not? Hey, we've got some good early voting. We're past 50%. Okay, we're at 100%. Good job. Okay, so the favorite answer was cell wall. Good job. You are correct. Good job. The next favorite answer was ADS ribosomes. If you pick that, that's okay. It's easy to get 70S and ADS backwards. Okay. But remember, eukaryotic cells, which include fungi and animals, are bigger than bacterial cells. We've got more stuff packed in our cells, so we've got to be bigger. So we have bigger ribosomes. Okay, so bacteria, 70S, fungi, animal cells, plant cells, protozoa, all ADS. Okay? And then the last one was endoplasmic reticulum. Actually, all eukaryotic cells have an endoplasmic reticulum. And that's my story, and I'm sticking to it. Because, you know, there's a few exceptions, but you don't have to worry about it. Okay, good job on that one. Let's go to our next one, which is the following is a structure that fungi have, but bacteria do not. Okay. Now this one, there's more than one right answer. Okay, so now we're thinking about the difference between fungi and prokaryotes, bacteria. Okay, we're getting some good early voting. Almost to 50%. There we go. Okay, we're at 81. We can make it to 100. We can make it. Almost. I just need one more. And just for the sake of time, we'll stop at 90. Okay, favorite answer, chitin in the cell wall. Good job. Okay. If you picked peptidoglycan in the cell wall, you just got bacteria and fungi backwards. That's easy to do at this point. Okay. So I wouldn't feel bad about it. But do know bacteria have peptidoglycan. Fungi have chitin. Okay. The next favorite answer is the nucleus. You're correct. Fungi have a nucleus. Bacteria do not. Okay. Next favorite one, ADS ribosomes, also correct. Fungi have ADS ribosomes. Bacteria do not. They have 70S. Okay. And then we have C, electron transport chain complexes. You know, fungi do have that. Okay. Some bacteria do, some don't. Okay. So, not the best answer, but sure, I'd allow it. Yes, Michelle.
Now I'll start out. I got this one wrong, but if this was on a test, would D be better than B because that's more of a structure than just what's in the cell wall? Because sometimes you throw those in there.
Yeah. If I
and I
asked
miss them.
understandably. No, for this one, I would make it a multiple answer question. Either that or I wouldn't have, you know, chitin in there with nucleus. If I did it by mistake, I would make it so that there were two right answers.
Thank you.
Oh, you're quite welcome. But that was a good question. Valid question. Okay, so we already talked about this one. Okay. The main ones that I want you to know for this upcoming test is the class of antifungal drugs that target ergosterol. And the example I want you to know is amphotericin B. Okay. So let's move on to our next question. I think I got somebody knocking at the door, but I think I'm going to ignore it. Okay, so over-the-counter antifungal drugs tend to be topical ointments. Why is that? Oh, I got to change this. Excuse me. Let me change this to a 4-question or 4-answer option. Because I've only got 4 answers here. Okay, we've got some good early voting. We're at 90%. Let's see if we can get to 100. And I think we're stuck at 90. Okay, favorite answer. Fungi have very few unique structures, so oral antifungal drugs tend to have more serious side effects. Good job. Yep. Next favorite answer, most fungal diseases are skin diseases, so it makes sense to treat them with a topical ointment. Also true. Okay. And then A, yes, fungi are opportunistic pathogens. Okay. We don't have very many true fungal pathogens. We have a few, but not very many. So that's a true statement, but it doesn't answer the question. Okay, so you got to watch out for this. So this one is also a true statement, but it doesn't answer the question. Okay. All right. So let's go to a review of viral replication. Okay. Let's go back to the whiteboard. All right. So we're going to go through this kind of quick, and we're going to worry about animal viruses because, that's what we're interested in. All right. So what's the first stage of viral replication? What does the virus have to do first? I'm going to do a non-enveloped virus system. I know it's
Penetrate?
number one thing. You know what? Let's go ahead and put that down. That's the second thing.
Adhere.
Very good. Yes, adhesion or attachment.
Yeah, attach.
Adhesion, absorption. Those are all synonyms. And thankfully, they all start with A. Okay, so it's attached. Okay, with this particular virus, it does like a bacterial phage. It does its uncoating and penetration all in one whack. Okay, so we'll go ahead and put uncoating. That's where we get rid of the capsid. Okay, what does that have to do next? Before it can go back out into the wilds outside of a cell.
Seek and destroy and gain nutrients.
That is true. Yep, it does have to. Oftentimes it goes in and it destroys the DNA of our cells. Okay. But why does it do that? And it's not really eating anything. It's like a little machine. It does need energy.
To perpetuate itself, to help it get in deeper, help it be
very
more
good. Yes.
viral.
So it needs to make more of itself. Okay. So it needs to make some capsid parts. And it needs to make more spikes. I'm going to put a little knob on the end so we can tell the difference between the capsid parts and the spikes. And it's got to make some more of its DNA genome. Okay. What do we call this phase?
Assembly.
Oh, that's the next one. After we make our parts, we got to put them together. That's assembly. Yeah, synthesis. Synthesis is next, where we synthesize or make a bunch of the parts. Okay. So we've got to use some of the cell polymerase to make mRNAs. And we use the cell's ribosomes to read the viral mRNAs and make, we've got spike coming out here. We've got some capsid proteins coming out here. Okay. And we need to have DNA polymerase make copies of this. Okay. So we're using the cell's machinery, we're using the nutrients the cell has brought in to use to make energy, ATP, to make all these parts. And then they start snapping together in assembly, kind of like Lego parts. We never have baby viruses. But these capsid parts snap together and we stuff genomes in them, okay? Because if there's a mistake and we accidentally don't get a genome in, it's not really a virus. Okay, so what's the last step? Now that we've made a bunch of new viruses that are going to go out and try to infect the neighboring cell, what do we call it? I know it's been a long time since chapter 6.
Replicate.
The whole process is replication. What's a fancy dancy term for heading for the exit?
Release.
Very good, release. Okay. So somehow these guys have got to get out, okay, so that they can infect a neighboring cell. Okay. Now, as we learned in the previous chapters on immunology, okay, our cells have ways of fighting this. Okay. We've got natural killer cells that look for stressed cells that are making a bunch of viral parts instead of their own proteins and what have you. And so they kill them before we can get to this release phase. Okay. And you remember the part, okay, we didn't go over this a whole lot, but when a cell gets infected by a virus, it will produce interferon. And it will release interferon. And that's basically a, and I left out quite a few letters. Sorry about that. So it's basically a cytokine, okay, a cell signal, chemical signal, that says to neighboring cells, hey, I've been infected, save yourself. Okay. And what that does is when a cell gets that signal, the interferon, okay, comes in, signals the cell to turn on different genes that if a virus comes in, it shuts down the action of the ribosome, okay, and the cell stops making proteins, okay. Sometimes it can signal the cell to die, taking the virus down with it and limiting the infection, okay. Cool stuff, right? Well, when we talk about coming up with antivirals, antiviral drugs, we have a hard time because so much of this would have to shut down the cell itself, okay, and kill the cell. And there's a limit to how much of that you want to do to your patient, okay. So antivirals, we tend to have a whole bunch of different drugs that act a different way. And we don't have nearly as many antivirals as we do antibacterial drugs, but we're getting more and more all the time, okay? So for the antivirals, okay, let's talk about some specific categories, okay, modes of action. And then there's, well, I say that there's only one I want you to know, really there's two of them. One of them is interferon, okay? Interferon is a natural product of your cells, okay? What we've done is we've taken the gene for interferon and we get little bacteria or yeasty beasties and factories to make it for us, and we pull it out and we purify it, okay? So we can use interferon to treat viral infections. Okay? If I understand correctly, the new very good treatments for hepatitis C that completely cure patients that have chronic hepatitis C. We give them doses of interferon, and it shuts down those infected cells and takes them down and takes the virus down with it. Cool, right? By the way, they also use interferon in some cancer treatments, okay, because it kind of does the same thing. Shuts down protein synthesis in cancer cells, which shouldn't be dividing, and kill them. Yeah. Okay, so interferon is an example of kind of an all-purpose antiviral. Okay. Doesn't work with all of them because some viruses have virulence factors that they can get around interferon. They either shut down interferon, they break it, or they can shut down the antiviral proteins that shut down ribosome synthesis, so they take out that. So anyway, depends upon the virus. Okay. Oftentimes we target enzymes that are specific to the virus. Okay. So, especially if we're talking an RNA virus, we do not, we don't make enzymes that make an RNA copy from an RNA template. Okay? We just don't do that, okay? Because that never happens in our cells. So the virus has to bring its own enzymes to do that. So we have antiviral drugs that specific viral RNA polymerases. Okay? When we're talking HIV, there are enzymes that will, HIV is an RNA virus. It has a funky enzyme called RNA reverse transcriptase, okay, that when the RNA genome comes in, reverse transcriptase does the reverse of what our cells do. Okay, it makes a DNA copy of RNA. Okay, we only do it the other way around. We only make an RNA copy of a DNA template. Okay? But reverse transcriptase will do that, and it'll make a double-stranded genome, okay? And then it gets sent to our nucleus, and it gets integrated or put into our genome, okay? Kind of like a prophage when we're talking about temperate bacterial phages, okay? Except for it's not temperate for us, you know, it causes nasty disease, and we would very much like to get that genome out, but we haven't figured out how to do that yet. So we can target the enzyme that does that. It's called integrase. Okay? So we tend to target enzymes that are specific to a virus. Okay? Or we will target a process, okay, that is necessary to the virus, okay, that we can shut down temporarily, okay, in a cell that's infected. Okay? So like I said that I wanted you to know interferon. I also want you to know another very common antiviral, okay, it's called acyclovir. And most antiviral medications have vir in the name, makes it easier to pick it out. Okay? Acyclovir is mainly used for herpes virus family infections. Okay, so if I get a cold sore, I can get an over-the-counter medication that contains acyclovir, or something that used to be, you know, a kind of a second generation acyclovir. It has cyclovir in the name, and put that on that, and it will shut down replication of the virus. It won't cure it, but it shortens the duration of that cold sore, and I'm less likely to spread it to other cells, okay, or to other people. Okay. Also, you can take it orally if you've got a bad case of shingles. Shingles is caused by a virus in the herpes virus family. So tends to have very few side effects. You got to watch out for it though. But acyclovir is actually a nucleotide analog. Okay, so let me tell you what I mean by that. So just as a reminder, nucleotides have a sugar group, okay, and they have a base. We'll go ahead and put this A here for adenine. Okay, and then we've got this sugar or this carbon sticking up. And then we have a phosphate group. Whoops, let's do that this way. Okay. And then I've got an OH here. And the plan is that I would bring another nucleotide in here. And just for the fun of it, we'll go alphabetically and make that a C. And we do a dehydration reaction. I left out a hydrogen here, which makes it a little more difficult to show the dehydration reaction. So I take this out and I hook this up. Okay. And I do the same thing as I go along. Okay. So a nucleotide analog is something that looks kind of like this, but it's not quite right. Okay. And so acyclovir looks kind of like this. It's got a phosphate group here. Okay. It's got a base, and I forget what the base is. We'll just put a B for base. Okay. So it can hook up here, but I don't have an OH group to hook another nucleotide to it. So it stops DNA synthesis. Okay. So the reason why it has very few side effects is, okay, the virus is making DNA when the cell normally wouldn't. Okay. So it's going to target the viral DNA and pretty much leave the cell DNA alone. Okay. Potentially, you could have problems with rapidly dividing cells that should be rapidly dividing, you know, like your skin, the tissues of your gums. Okay. But acyclovir is very, very good at doing this, and it has relatively low side effects. Okay. Cool stuff, right? Hey. Oh, and another one I forgot, in the category of natural products, okay, monoclonal antibodies. And I bring that up because it's been effectively used, monoclonal antibodies have been effectively used for folks that have gotten severe cases of COVID, okay? Before, what we would do is we'd get folks who had had COVID and got better, we'd take their blood, separate out all of the antibodies, okay? And generally that would be called a polyclonal antibody if we put that into somebody else's veins, okay? Because usually it's kind of hard to separate out which antibodies are against which, okay? But what we can do is we can get, I won't go into the whole process, but you get immortal cells, okay? And you fuse them with a plasma cell, okay, that's pumping out the antibodies, okay, and we figure out, you know, oh, these are antibodies against COVID, so we call these anti-COVID or SARS-CoV-2 antibodies, okay, and we fuse them, and this immortal cell makes it so this plasma cell doesn't die. And so in factories, we can make a ton of very, very specific antibodies, okay? And so then we take these anti-COVID antibodies and deliver them to a patient that is suffering a severe case of COVID, and they get better faster. They're less likely to pass away. Okay, questions? I know I threw a lot of mechanisms at you for the antivirals, but because there's so few targets, we have lots of different ways of trying to shut them down, hopefully without causing too many side effects.
So Teresa, what about lysine? Do you ever recommend that to people?
I haven't heard of that one.
Okay.
What's it called?
It's lysine. It's a natural supplement. And the second you think you're
Oh.
going to get a cold sore, you start taking that and you will never get a cold sore. And I've
I
recommended
actually
that
have
to
heard of that.
people
Yeah, I
that
have heard of that.
they get herpes breakouts. If they stay on lysine, they don't get their, little friend every now and then, like
Mhm.
they would without taking it. So that's a good natural,
Yeah, see, I don't remember what lysine has in it.
and
Now
it's
I'm
just
curious.
it's it's just a supplement, but it's L-lysine, and just all natural.
You know what? Don't quote me, but I think that's an amino acid.
Yes, it is.
Yeah, and I think, don't quote me on this. I used to know this, but it's been a while since I did my work on herpes simplex. I think what it does is it binds to the spikes.
Mm,
I think, and
Okay.
it's an amino acid that we need, that we
Yeah.
use, but I think it just kind of fits into kind of that docking station.
oh,
Or it might do it on the receptor that the herpes is trying to grab a hold of.
okay.
Don't quote me on that. But yeah, no, I've heard that that's a really good supplement to take if you do have a lot of cold sores.
Yeah, back in the 70s and 80s when I laid in the sun all the time to, get my tan, I always got the sun sores on my lips, and somebody recommended that to me. And the second I feel a cold sore, man, it just takes it. I don't even get it. I don't even get it. I don't lay in the sun anymore, but you know what I mean?
Oh, no, I know what you mean. Yeah, because that's one of the triggers. It depends upon the person, but yeah, exposure to UV can trigger
Yeah.
herpes to come out of hiding.
Yes.
But yeah, no, I'd forgotten about that one.
Yeah.
I used to know the mechanism of that one, but yeah, no, see, there's all sorts of stuff, but it's just kind of like something here, something here, something here, and it's usually specific to the virus.
But
So
to know,
yeah, L-lysine probably would not work for COVID. It might, but it might not.
right. But to know how they work now, I mean, to me, if a person like taking your class, now you can visualize, oh, that's how that works, because otherwise you would think, oh, how could that work? How could that? But when
Yeah.
you really have a visual of what it's specifically doing or targeting or how it's targeting, it's really makes it more believable and interesting,
Mhm. Yeah, it's pretty cool. I mean, I do not like, I've never enjoyed any virus I've gotten. But they sure are fun to study, because they're just like evil little machines, and who doesn't like evil little machines as far as studying them? I don't want to get them. So all right, let's go on to our last topic before I ask you questions. And I'm suppressing an evil laugh. Let's talk about antimicrobials in general and how the pathogens can develop resistance. Because as we all know, there are bacteria that are resistant to antibiotics, and they are causing a problem. So let's talk about the very broad mechanisms for how a pathogen would develop resistance to an antimicrobial. Okay, and we're going to talk about bacteria because they're easier to talk about. But the same kind of thing applies to fungi and protozoa and to viruses. Okay, so there are some basic ways that pathogens can develop resistance. Okay, and I'm going to use some specific examples from the antimicrobials I want you to know. Okay, well, let's start with penicillin. Penicillin's been along for a long time. And we have an awful lot of bacteria that have developed resistance, okay, to penicillin. And what happens is in the normal course of things, penicillin is imported in and it shuts down the enzymes. Okay, let me go ahead and get my cute little Pac-Man enzyme. Okay, that make peptidoglycan. So that's the goal. So they stop making peptidoglycan. We start getting holes in the cell wall and then the bacterium lyses. Okay, that's the goal. Well, resistant bacteria, let's go ahead and put penicillin here. Penicillin is in the beta-lactam family of drugs, and it's got this circle, okay, that we call the beta-lactam, and it's attached to the rest of the molecule. But resistant bacteria have a gene, okay, that makes an enzyme that we either call a penicillinase or a beta-lactamase that comes through and it goes snip, okay? And so this, I'm going to call it a beta-lactamase. Okay, chops up the drug, okay? So that is the first one that I want you to know. Inactivation of the drug. Okay, so they've got some sort of enzyme that goes snip, 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 snip. Okay. Now that's the most common form of resistance to penicillin and the other psyllins. Okay, so penicillin became less effective, so we made other types of psyllins that have a molecule that kind of protects this beta-lactam ring, part of the structure that protects it, and then we get a mutation here and we get another beta-lactamase that can get past that and still go snip, snip, snip. Okay, so it's kind of like an arms race. We come up with something different. There's a mutation that gets around what we've changed. And so we eliminate the susceptible members of the population, those that are resistant, divide, multiply, take over the population, and then we've got to switch to another drug. It's an ongoing thing. Yay, evolution. Okay, changes within populations. Okay, now the other thing, okay, that can happen. Okay, let's talk about tetracycline. Okay, remember that tetracycline goes in and it will bind to 70S ribosomes. Okay, shutting down protein synthesis. Okay, so the other thing that a bacterium can have happen is it gets a mutation. Okay, so here's the gene for the ribosomes, and I get just a little bit of a different shape of ribosome. It can still do its job, but now tetracycline has a hard time grabbing hold of it. Okay, so we call that alteration of the target. Okay, so we get a little bit of change in the ribosome so that tetracycline doesn't bind to it as well. We get a little bit of a change in the shape of the enzyme that makes peptidoglycan. Okay, so penicillin can't mess with it. Okay, we get a little bit of a change in the shape of the enzyme that sulfa drugs compete with. Okay, so yeah, that makes the bacterium resistant. Okay. The other thing that can happen, okay, is they can change their transporter genes, okay, where You know, they can either decrease the number, okay, of transporter proteins, okay, or they change the shape so that the drug can't get in. Okay. So we call that limiting entry, okay, or transport. Okay. Okay, the last one we're going to talk about. And this one is a problem. Okay. Oh, and let's go ahead and let's show an antibiotic, you know, just, can't get in, so it doesn't get in. Another one is some bacteria have a fancy transporter protein that we call an efflux pump. And basically, it picks up anything that's of hazard to the bacterium, okay? And it goes, nope. And as soon as it comes in, okay, so penicillin got in here and the efflux pump says, nope, out you go. Okay. So with these efflux pumps, pump the drugs right back out. Okay, so it reduces the concentration of the drug inside the cell. And a lot of multi-drug resistant bacteria, superbugs, okay, have an efflux pump. So it's just like, oh, you're bad, out you go. Okay. Oh, and I forgot one of them. I said that was going to be the last one. Sometimes if, you know, like if I'm exposing a bacterium to penicillin, okay, or tetracycline or sulfonamides, okay, sulfa drugs, these cells will respond by overproduction of the target. And since the target is usually an enzyme, I'm going to put targeted enzyme. Okay. Yeah, I forgot about that one. Okay, questions about mechanisms of resistance. Okay, so these are the ones that we've seen in bacteria, okay? But let's go ahead and talk about viruses real quick, okay? Viruses can do this one. They can alter the target, okay? They can get a mutation in their enzymes, the viral specific enzymes that we're targeting, okay? And the antiviral either is less effective or it's not effective anymore, okay? Or if we're talking about L-lysine or about spikes on COVID, okay, that are the targets of monoclonal antibodies, whether we're making them because we've been vaccinated or we're getting monoclonal antibodies through an IV. If those spikes are too different of a shape, those antibodies aren't going to work anymore. Yep. Yeah. So this is generally what we see, okay, in viruses. Is alteration of the target through mutation. Okay. Occasionally you can get a mutation where you get extra copies of an enzyme. So occasionally we'll see that over here, but not very often. Okay. So with viruses, they get these mutations, spontaneous mutations that happen, or they can pick up new genes through reassortment. Okay. I want to remind you about how bacteria share antibiotic resistance genes. Okay. They do that through horizontal gene transfer. Okay, so yes, you gotta go back and you gotta review that. Oops, I just got rid of my peptidoglycan cell wall here. So yeah, review horizontal gene transfer. Yes, you will need to know the infamous transes. Okay, transduction, transformation. Okay, and then there's conjugation, which is not one of the infamous transes. Okay, but that's how they share these genes. And that's besides having a mutation show up here, and then this cell passing the mutations on to its offspring, they can also share genes across unrelated individuals. We wish they didn't because it's annoying, but that is also how resistance spreads. Okay, questions about that? Okay, I think I've got time to ask you 2 questions. Let's see, let's go to, let's skip this one. Let's skip that one too. Okay. Let's get a poll. Let's relaunch the poll. Yay! Acyclovir is an anti-herpetic or anti-herpes drug, which is a nucleotide analog. Which of the following enzymes or structures would be targeted or affected by acyclovir? Okay, we've got some good early voting. Okay, we're at 72%, 81. Well, give me 90. There's 90, 100%. Good job. Okay, the favorite answer was DNA polymerase. Good job. Did a very good job. You picked one of the others. Don't worry. By struggling to remember, by picking an answer, you're going to remember the right answer better. Okay. All right, let's go to the next one. Okay. This one talks about influenza. So after tricking the host cell into engulfing it, that's part of how influenza virus does the penetration and well, attachment and penetration. The influenza virus fuses its envelope with the membrane of the endosome as the pH drops. Okay? So it's been phagocytized. It's getting ready to fuse with the lysosome. It's intending to break down the influenza virus, but the influenza virus uses that drop in pH to get out. Okay. So Tamiflu is an anti-influenza drug that prevents fusion of the envelope with the endosome. Okay. So it stops that stage of uncoating. Okay. Which of the following enzymes or structures would be targeted by Tamiflu? Okay, we've got some good early voting. And we're at 54%. And by the way, I need to fix this. This should be say endosomes and not lysosomes. Fix that one. Okay, we're at 90%. See if we can get 100. Now, for the second time, I'll go ahead and end it. Okay, favorite answer was pH pumps and lysosomes, even though it should have said endosomes. Good job, you got the right answer. Okay. you picked one of the other answers, considering that I had the wrong word there, I can understand going, no, it can't possibly do that. Question asked about enzymes. Okay, good job, everybody. Let's go ahead and go to what to do for Friday. Okay, so the only thing that's actually due is the CLO post assessment. That is due on Friday. You will get full points for all the answers you get correct. For the answers that you get incorrect, I'll give you 75%, 3/4 of the points. Okay. So in other words, I want to inspire you to do your best, but I don't want to stress you out too much. And it is timed, and I don't give you time to look up the answers. I want to know what's in your head. Okay. I'm going to take that as an average for the class and compare that to the class average at the beginning of the semester. And if I see that there's a course objective, learning objective, that isn't as high as I would like, I know to emphasize that a little more or a little differently next semester. So this helps me improve the course. Okay. It's not really due by Friday, but I would do it by Friday. If 80% of the class does a course evaluation, then for those who did it, I will give you 3 extra credit points. Okay. You have until Monday at 8 a.m. So effectively, you have until Sunday to do it. You can turn in the screenshot of where it shows your courses and which evaluations you've done. You have until the end of the semester to do that. But you have until Monday at 8 a.m. to take the actual course evaluation. And then also another extra credit opportunity that is due until the end of the semester. CWIT wants at least 75% of the students to take the cybersecurity badge. Okay. Yes, sure.
What did we decide we're going to do about that? I tried to go in this morning and it's still the same as, I think it was Lily that mentioned that the other day. It's still just one screen of reading and it says you're done.
And you know what, if you read through it, you feel like you know what you need to know about cybersecurity, that's good. Just take a shot of the badge. But if you want more, if you go in and click on it, you can take the training as much as you want. If you click the other option other than text, it has some videos and stuff that walk you through it.
So you don't actually get the badge because it doesn't let you go through the motions of getting the badge. Like I read it, it says you're done. I went to my badges. There's nothing, there's no action unless someone else has found something different, but
maybe you have to watch the videos.
I did.
No,
You
you
have to watch the
don't.
videos and go all the way through.
You don't
And then
have
you'll
to
get
watch
the badge.
You don't have to watch the videos.
You
So
just
what do you,
have to do the reading, and then it should, once you exit out of doing that reading screen and you go back into your badge book, mine was all the way at the very bottom. It was the last badge and it just said that you've completed the cybersecurity badge, but all I did was the reading.
Yeah, mine came up on the screen after I was done reading. In fact, I had to do it twice because I missed it the first time.
and you did the text option?
Yeah.
Huh. I'll try again.
There's something you say down at the bottom that says you're done.
Uh-huh.
And I did something like that, and then something popped up that said, congratulations, you're done.
Okay, thank you,
Yeah.
gang. I'll try again.
And then if it doesn't work, put in a help desk ticket.
Okay.
But for the assignment, go ahead and take a picture of where it says, yeah, you've completed it. As long as I have some sort of verification. So thank you. Thank you to those who have done it both ways and have had it succeed. So thank you. Okay, so here's the plan for next week. As you know, next week is finals week. So we do not have any new material. I am done presenting new material. So you are not required to attend lecture next week, but I will be here during lecture time. And I'm happy to go over any topics you want to go over It's kind of like a study session slash office hours. Okay. So that is the plan for next week. Completely optional. Okay. Also, that's a good time to finish up any extra credit opportunities. Okay, like submitting the screenshot for the course evaluation or your screenshot for the cybersecurity. And then exam 5 is available starting Friday and through a week from Saturday. Okay. And this exam is part of the ones that I will drop the lowest score. So if you're happy with your exam scores, you don't have to take it, but I would recommend taking it just because, I don't know about you, but I study better and more effectively if I know I'm going to take a test. I remember it longer. Okay, any questions about the plan for next week?
What did you say, come to class and it's going to be like a study session or
Yeah.
different time? Okay, so normal time for class.
Normal time. Yep, normal
Okay,
time for class.
Just no new information. Okay,
Yeah.
all right.
In fact, I'm doing that. I've kind of modified, what I'm doing as far as office hours, because I figure it's easier for people to come at the time that they're normally scheduled. Okay. And then there was the suggestion that I offer extra credit for coming on Monday. I'm at the maximum allowed extra credit that the department will allow. They put a cap on the percentage of extra credit points based on the total points. So it would be wonderful if we could do that. I know that's terrible. It's terrible that the department limits the amount of extra credit, but I'm a team player. They ask me to do stuff, I do it.